everyone. I'm Sakshi Gupta, faculty in Life Science Department in Bansal Academy. With me in this video, we are going to discuss the questions related to unit of animal physiology from CSIR Life Science exam held on November 21, 2020. We are going to discuss questions from shift 1, that is morning shift. So the first question from animal physiology in section part B is question number 27, which is myosin molecules that assemble into bipolar filaments in the muscle are. The options include myosin 1, myosin 2, myosin 4 and myosin 5. So guys, the muscles in human body, basically they have got the structural and functional unit known as sarcomere, in which you have got thin myofilament and thick myofilament, the filament which exists in the structure of muscles. The thin myofilament, they comprise of actin microfilaments, while the thick myofilament, they comprise of myosin protein. Myosin protein is basically a motor protein which, by utilizing energy from ATP, converts the chemical energy to mechanical energy, which in the case of muscles is ATP being converted to contraction. So to assist this, the myosin protein, it forms cross bridge with the actin microfilaments, and there it assists the movement, the process which we term as muscle contraction. Alright, so the myosin protein in case of humans, it is subdivided or it is classified into total of 18 families, which are classified as myosin 1, 2 and so on till 18. So the options that has been given to us in our question, if we deal with them one by one, then of the muscle protein available, the myosin in that, in case of muscle, the myosin which exists is type 2. This myosin is the most conventional myosin which exists in the human body in case of muscle cells, and this is the most sterid one. The myosin 2 protein, this forms a cross bridge with the actin microfilament, and this is the one which will be involved in the process of muscle contraction, where it will convert chemical energy to mechanical energy to execute this process. This myosin 2, this is the one which is most abundant in case of human body, which exists mainly in the muscle cell. It also exists in non-muscle cell as well, but there it exists as transitory structure. Transitory means it won't be permanent. ये कुछ टाइम के लिए होगा, and then जब इसका काम complete हो जाएगा, तब उसके बाद it will be lost inside the non-muscle cell. While in case of muscle cell, this will be a permanent structure. So in this case, the correct answer is myosin 2, which is found in the muscle cell. Other than that, myosin 1, this is the one which is another ubiquitous myosin in case of humans. Ubiquitous matlab it will exist in all the cells. But this is the one which is involved in trafficking of vesicles, right? Myosin 5, this is again the one which exists in case of humans. This is involved in trafficking of vesicles, Golgi, ER and other organelles. It means in their shifting in their movement. Myosin 4, this is the one which has got a little bit of unconventional structure, unlike other myosins, right? So of these, the one which exists in muscle includes myosin 2. So our next question is question number 34, which is which of the following signaling molecule is not a protein or a peptide? And the options are transforming growth factor alpha, erythropoietin, granulocyte colony stimulating factor, and epinephrine. So let's discuss all of these one by one. First of all, transforming growth factor alpha. This particular hormone is proteinaceous in nature. Alright, it is a trimeric protein which is secreted by immunologically active cell, such as your activated macrophages, your activated lymphocytes such as NK cells, neutrophils, TH cells, etc. And regarding their function, these are the ones which are involved in inflammatory response, your systemic inflammatory response during immunological reactions. Right? So this is proteinaceous in nature. This eliminates this option. Then erythropoietin. This is the hormone which is glycoproteinaceous in nature. Right? And this is secreted by kidney cells under the hypoxia condition. Hypoxia when the oxygen level is low. Right? So in that condition, kidney secretes erythropoietin hormone, and this is basically responsible for erythropoiesis, which means erythro means RBC, poiesis means synthesis or formation. So this is involved in formation of RBC. So this hormone is glycoproteinaceous in nature, hence this option is also eliminated. Right? Then granulocyte colony stimulating factor. These are the hormones or cytokines which again are glycoproteinaceous in nature, and these are secreted by the cells which allow the migration of HSCs, which is hematopoietic stem cells, to blood from bone marrow, right? And other than that, these cells, this hormone GCSF, this also allows proliferation and differentiation of granulocyte, and these are also secreted by your immunological cells, right? Other than that, so this also eliminates this option as well, right? Then epinephrine. Epinephrine, which is also known as adrenaline hormone, this is the one which is secreted by adrenal medulla portion of adrenal gland. And chemical wise, these hormones, epinephrine or adrenaline, these are catecholamines which are derived from amino acid tyrosine. So these hormones, they are amino acid derived hormones, right? So they are neither proteinaceous nor of peptide nature, right? So catecholamines, these are derived from tyrosine amino acid, epinephrine hormone, and this is involved in your triple F response, that is your flight, fight and fear. Okay, fright, fight and fear. Hence the adrenal medulla is also referred as triple F gland because of this nature, where during emergency situation or during adventurous condition, this hormone is secreted, and this results in increase in heart rate, then increase in carbohydrate metabolism, increase in blood pressure, causes vasoconstriction, and other effects which occur during the adventurous or during triple F condition. Right? So amongst all this, epinephrine is the one which is amino acid derivative. Right? So this gives us with our answer epinephrine. While rest the three options, they are either proteinaceous or glycoproteinaceous in nature. Right? So the answer for this question is 4. The next question is question number 45. Which one of the following causes constriction of blood vessels? Which means which one of the following is a vasoconstrictor? So the options are carbon monoxide, nitric oxide, endothelin 1 and atrial natriuretic factor. So let's discuss them one by one. First of all, carbon monoxide. This is the one which is secreted by capillary endothelial cells and their function is that they cause dilation of blood vessels, that is, they are vasodilators. The carbon monoxide, this is the one which acts as neurotransmitter as well as hormone in case of nervous system. Right? So this one is of nitric oxide, secreted by capillary endothelial cell and causes dilation of blood vessel. The other one is carbon monoxide, which is neurotransmitter and hormone in case of nervous system. Both function as vasodilator and causes dilation of blood vessel. Right? So this eliminates option 1 and option 2. Okay. Now the third option, endothelin 1. This is again secreted by capillary endothelial cells and these act as natural antagonist of NO and CO. These are natural antagonists of nitric oxide and hence these function as vasoconstrictors which causes constriction of blood vessels. So this gives us our answer endothelin 1, but before checking it out, let's check the other option, atrial natriuretic factor. Atrial natriuretic factor, it is secreted by heart atria in response to atrial stretch condition. The ANF hormone, which is secreted by heart atria, it acts on the kidney cells and there it causes sodium excretion and it also causes dilation of blood vessels in the glomerulus. Right? So this is again a vasodilator and this functions in antagonist manner to the enzyme cum hormone called renin from your RAS system. That means renin, angiotensin, aldosterone system, which is responsible for causing vasoconstriction. So ANF itself is a vasodilator which is natural antagonist to renin. So amongst all the options, the NO-CO, they both are vasodilator as well as ANF, which is again a vasodilator. The endothelin 1 is the vasoconstrictor. So the correct answer is option 3. The next question is question 46. Which one is not the function of P cells in the collecting duct? Options are sodium reabsorption, potassium secretion, water reabsorption and proton secretion. So coming on to the solution of it, your collecting duct, they are the part of nephrons, which are the structural and functional unit of kidney. So collecting duct comprises of two different types of cell. One is P cell, which is also known as principal cell, right, or light cell, and the other cells are intercalated cells. So here we will discuss about the function of the P cells, which are actually most numerous in the collecting duct, and coming on to their function, they are involved in salt transport and water transport. So first, for salt transport, the P cells, they have got specific cells, specific transporters in their cell. First transporter is ENAC, which is epithelial sodium channel, and the second involves K positive channel. These two channels, they exist in the membrane of the P cells, and under the influence of the hormone aldosterone, the ENAC channel, this is involved in sodium reabsorption, right, and via K positive channel, they are involved in K positive secretion. So that's how they regulate salt transport, right, and thus our option 1, that is sodium reabsorption, and option 2, K positive secretion, both are the function of P cell, and we have been asked which one is not, right. Coming on to the second transport, which is water transport. The P cells, they have got water channel aquaporins, which is AQP2 in the membrane. AQP2 channel, under the influence of hormone ADH, antidiuretic hormone, or also known as vasopressin, through aquaporin 2 channel, the P cells, they are involved in water reabsorption to conserve water inside the body. So that's how they are also involved in reabsorption of water, and via these, the P cells in collecting duct, via these mechanism, they are involved in maintaining extracellular fluid volume, and also by regulating salt balance, they regulate BP. So amongst the given options, sodium reabsorption is the function of P cell, K positive secretion and water reabsorption, but they are not involved in H positive secretion. Hence option 4 is our right answer for this question. So the next question is question number 47. Which one of the following is not produced by alpha amylase dilation of ingested amylopectins? Now amylopectins, these are basically the part of the storage polysaccharide, that is starch in case of plants and glycogen in case of animals. Both of these, they are homopolysaccharide which has glucose as the monomeric unit and linked by alpha 1,4 glycosidic linkage, right? The starch and glycogen, they comprise of two units. One is amylose and other is amylopectin. The amylose is the straight chain part, while the branched part is known as amylopectin. So on this, if enzyme alpha amylase act, the enzyme alpha amylase is basically endoglycosidase, which acts upon the glycosidic bond but internally, right? Not on the terminal glycosidic bonds. And this is alpha 1,4 glycosidase in nature. So digestion by alpha amylase on amylopectin is going to yield you a disaccharide unit, maltose, which is a disaccharide. You can have trisaccharide, that is maltotriose, which is trisaccharide, and both comprise of glucose unit, this of 2 glucose unit which are linked by alpha 1,4 glycosidic bond, this by 3 glucose unit linked by again by same alpha 1,4 glycosidic bond, and you will have alpha limit dextrins, which are small part of branched amylopectin, which consists of few glucose unit linked together, right? So you, after digestion by alpha amylase, you will get the disaccharide, trisaccharide or the oligosaccharide unit, but never its monosaccharide unit. To get the monosaccharide unit, these will be further digested by disaccharidase, for example maltase, right, or by dextrinase, which will further yield glucose as the final monomeric unit, which will complete the digestion of the starch or glycogen, including amylose and amylopectin. So action by an endoglycosidase, which is alpha amylase, is going to yield maltose, maltotriose, alpha limit dextrins as shown here, but it will not yield glucose molecule. This is the one which will not be yielded by the action of alpha amylase. Hence, the correct option for this question is 1. So next question in animal physiology is from part C, question number 110, which is related to testosterone. Testosterone is a steroid hormone about which a set of statements are given below. First, it is secreted by Leydig cell of testis. B, it is secreted by Sertoli cell of testis. C, it inhibits secretion of luteinizing hormone from anterior pituitary. D, it does not inhibit secretion of follicle stimulating hormone from anterior pituitary. Which of the following combination represents correct statement about the testosterone? And first option is A and C, second option is B and C, third option is B and D, fourth option is A and D. So to choose the correct statement, first we will discuss the A and B statement, which deals with the Leydig cell and the Sertoli cell of testis, right? So testis, it is the main male reproductive organ which has got seminiferous tubule as its structural unit, right? The seminiferous tubule, this is the one which as the endocrine part consists of two different type of cell. One is Leydig's cell and the other is Sertoli cells. Leydig cells, they are also referred as interstitial cells, and the Sertoli cells, they are referred as subtentacular cells, right? So the hormonal secretion of the Leydig cell is testosterone, which is the main male androgen, the one which involves the development of primary and secondary sexual characters in males, and also supports the process of spermatogenesis, that is formation of sperm, right? And Sertoli cell, the hormonal secretion of these cells include androgen binding protein, and as the name suggests, this binds the androgen, male androgen, which is testosterone only. This is your androgen hormone in case of male. So this is the one which binds testosterone and concentrates it in interstitial space, right? So that testosterone is available for the process of spermatogenesis. And the another hormone which is secreted by the Sertoli cells includes inhibin. These are the mother cells or nurse cells which are involved in providing the nourishment to the Leydig cell, right? So as we can see over here, the testosterone is secreted by Leydig cell and not by Sertoli cells. So hence, amongst A and B, our A option is correct, A statement is correct, but B statement is wrong. Now, we will discuss about the hormonal regulation which has been asked in C and D statement. So, regarding the hormonal regulation, the hypothalamus, it secretes a hormone GnRH, which is gonadotropic release hormone. This acts on anterior lobe of pituitary gland, and from this it stimulates the release of luteinizing hormone, which I am writing as LH, which is also known as interstitial cell stimulating hormone, right, in case of males, and it also causes release of another hormone, follicle stimulating hormone, which I am writing as FSH. The luteinizing hormone, it acts on the luteinizing hormone receptors present on Leydig's cell in testes, and by this it stimulates the release of the secretion of testosterone. While the other cells, follicle stimulating hormone, it acts on the follicle stimulating hormone receptor present on Sertoli cells, and from them it causes the secretion of hormone which we discussed earlier, the androgen binding protein used for concentrating testosterone, and inhibin. So when the testosterone concentration is low, in that scenario, the hypothalamus, it goes to this cycle and results in the secretion of testosterone. And then the testosterone level, it gets high. In that case, the testosterone, it gives the negative feedback loop to inhibit gonadotropic release and release of luteinizing hormone. So testosterone and LH, they are present in the negative feedback loop. Whenever the testosterone concentration is low, During that condition, hypothalamus will release gonadotropic release hormone, which will act on pituitary gland to stimulate the release of testosterone via luteinizing hormone. And when the testosterone concentration is high, it itself will inhibit the release of gonadotropic release hormone as well as luteinizing hormone and follicle stimulating hormone. And also inhibin will also cause this inhibitory effect on this cycle. So coming on to the statement C and D, it inhibits secretion of luteinizing hormone from anterior pituitary. This statement is absolutely correct as discussed over here. And it does not inhibit secretion of follicle stimulating hormone. This statement is not correct. So hence, amongst the given statement, A statement as C statement is right. Therefore, option 1, which is A and C, is the correct answer for this question. So these are regarding the animal physiology question. To check question related to other units, please check other videos from the Bansal Academy. Thank you.